This, as I mentioned, if you have these tumors being driven by these proteins, which are fusion proteins, the treatment sometimes can be much better. And it does not have so much collateral damage in terms of side effects, because normal cells actually will not have these hybrid genes. So this has been used, and have a look at this uh, drug called imatinib, which actually it does exactly the same thing. It binds to this ABL kinase, and by doing so, it can actually block this kinase, ABL, BCR ABL, to function. And you can see the drug itself is super efficient. You can see before Gleevec, you see a big gastrointestinal tumor. And 1 month after the therapy, of course, if the tumor was driven by this kinase, you can see that the cancer is largely gone, and all you see is normal bladder. But the problem we face is the ability to transfer uh, this finding. This is not a representative efficacy slide, if you understand what I mean. This happens in a lot of patients. For some diseases, it's very rare that you will see extended efficacy of this sort. But for many orphan diseases where there is no treatment, basically nothing works. And please don't forget, many of these tumors are heterogeneous, so they have a tumor heterogeneity. Within the same tumor, some cells are being driven, the growth is being driven by some drivers, for example, driver A. Within the same tumor, there is heterogeneity, so within some cells are being driven by driver B, and there may be a third subpopulation which is being driven by driver C. So if you take a tumor sample and you don't take a representative tumor sample, you take part of it, miss part of it, you're only going to see, okay, it looks like only driver A is present. And based on this, the medical oncologist may start therapy targeting driver A. Well and good. After subsequent treatment biopsies, if they are taken again, it might look like, okay, the cancer is being driven by driver B and C. Well, this was the case from the beginning. What you have done by blocking driver A, you have allowed those cells which were being driven by driver A to disappear or decrease in number, and now the tumor is predominantly driven by even more nastier drivers, driver B and C perhaps. And that's why if you do sampling now, it might look like that actually there are now new drivers of the same disease. While we know most of the human cancers, adult cancers, are in fact driven, they have an endogenous inherent tumor heterogeneity. And that is a feature of cancer that makes it very difficult for medical oncologists, for the doctors, to treat cancer. So this was about the drivers, the oncogenes, some of the oncogenes, and they come in all shapes and forms. I discussed some of them with you. What about viruses? They can also cause cancer. And this is such an important discovery that a Nobel Prize was awarded on the discovery of the sarcoma virus of connective tissue, chicken sarcoma virus that causes cancer in chickens. And there was a very simple experiment done. Chickens would develop this sarcoma in the breast muscles, and it looks like this was infectious. So if you have this in populations, others will get it as well. It turned out that, so what Rouse did in the experiment done at Rockefeller in New York is take out the tissue, cancer tissue, and ground it up to make sure that you can actually get, if there are any agents which are causing cancer. Previously, it was not known that cancer are actually transmittable. It was thought that there may be genes underlying that is causing cancer. But it looks like what they did with in this experiment, use this make a very fine paste of these cancer cells and filtered them and getting a very clear supernatant here, nothing here, and injected them back into chicks, and identified as they grow that actually they formed also cancer. And that was one of the first very important reports showing that cancers can be transmittable. And what is driving in this case the cancer is again an oncogene. It's called SRC. You have seen in the textbook there are V and CSRC. We have counterparts in human cells, which is called CSRC, cellular SRC. But viruses also carry genes like SRC, which is a viral form. So you will have initials VCSRC in those cases. This is not virology lecture, so I'll keep it brief. But you can see here what happens when the virus goes in cells. This is an electron micrograph. They hijack the machinery of a host cell, and in doing so, sometimes they start producing their own copies in great numbers, which is a typical example of a lytic cycle. And you can see lots of new viruses are putting out of the infected cells. So this would be a classical example. Many viruses, how they cause such cancer, they carry lots of times these oncogenes, but sometimes they can actually stop the good genes in our body, tumor suppressors, which I'm now going to start. They can stop the good tumor suppressor genes from functioning, from saying there is something wrong with cells. So you can see there are 3 viruses shown on this slide. There are actually many more. You can see here, this is SV40, and they have a specific proteins, in this case, large T antigen. Adenoviruses, proteins like, they don't cause cancer as such in humans, but flu is attributed to them. And the famous HPV, I know you can see they contain different proteins, E7, E6, large T antigen. Many of them are actually involved in blocking the function of good genes, which keep us safe, which we will start in a minute now. Uh, so viruses are, you can see, can give rise to transmittable tumors. The alarming thing is that we only know about a handful of such cases. There are reports that viruses particles have been found, viral DNA has been found by some in even prostate and breast cancers. Those studies need to be validated, but there is very limited extended data on such tumors. So it looks like that there may be, we are sitting on the tip of an iceberg. There may be other transmittable cancers attributed to viruses as well. So the other feature of the cancer would be that the good genes, they become active. We call them tumor suppressor genes. The cancer cells don't listen to good genes. In other words, they can evade growth suppression. So this is very important. The good genes, what I'm calling the tumor suppressors, are actually much more higher in function. They have an upper hand in deciding whether a cell is going to become cancer or not. And I'll tell you in a minute why this happens. So have a look here. These are the genes which are there in cells, but they don't get active all the time. Uh, are they only active in tumors? I'll show you that is not the case, but they are there to make sure that any cell which has an oncogene, uh, previously they were proto-oncogenes, if there are mutations and the cell takes a first step, perhaps by enhancing the rate in which they enter the cell cycle, these tumor suppressor genes can sense that activation of oncogenes. And they try to repair these defects in oncogenes, converting them back into proto-oncogenes, getting rid of them, the process called apoptosis. So as long as these are there, the tumor suppressor genes, we don't have to worry about developing cancer. And have a look why they are there. Are they only there in cancer? You can see in this embryo, there are genes, tumor suppressor genes, which are very important. So embryos do express genes like retinoblastoma, APC, but most importantly, the function of P53 is very well established in cancer. And what happens in apart from cancer is that they carry out a very important role in the development of embryo. Why? Have a look at this slide. What you're seeing as an embryonal hand, the development, and you can see here in this slide that there are these black dots. Can you see them? Basically, what it is telling you are the staining for the P53. So these are the points where P53 is becoming active. And why this is needed? If P53 will not become active, we will not get our hands. We will not be able to write anything, document history, and make uh, arts and so on. So you can see even for the development of embryo, the tumor suppressor genes are so important because their major function is in carrying out, in this context, is to carry out apoptosis and remove these barriers between the fingers that lead to us having these nice beautiful fingers. Without them, that would not have been possible, and we would have ended up having webs. So what happens in cancer is that these genes sadly are mutated as well. And when they get mutated, this time it will not be a gain of function like happens in oncogene. This will be a loss of function. So sadly, they will not be able to function properly, and that can cause cancer. But you can see equally, they're very important for the proper development of the embryo. So there are some questions. I'll take them in the last 15 minutes. Please feel free to, well, Dr. Aziz has also answered, but yeah, please feel free to leave them until the last 15 minutes, and then I can take those which remain unanswered. So you can see here how important these genes are, and how do they become active? You can see that they can cause massive apoptosis. They can cause massive cell death in embryonal stages. Of course, this is needed for metamorphosis, for the development of, in this case, proper hands, and similarly other organs as well. So how do these genes become active? There are lots of things which can activate P53. You may see in slides, and this is one of the exceptions, that P53 is the name of the protein, but the gene for P53 is called TP53. Usually you have the same name for gene and protein, but in this case, the gene that codes for P53 protein is called TP53. So have a look what happens. Any signal, in a sense, that leads to the damage, that poses a threat to the DNA, can actually activate P53. And when it becomes activated, I was giving you an example of epidermal growth factor receptor. What happens is that P53 needed in order to activate the receptor. Same was the example of androgen receptor. You need the dimer to bind to the DNA in order to drive transcription. In this case, it's not a dimer, it's a tetramer. So you need four copies, essentially identical copies of P53 protein to come together to form this active protein. And therefore, you need a lot of different, you can see here different signals are needed. And you can see P53 can be activated by lack of nucleotides. What will happen if you don't have nucleotides? Well, they are the building blocks of DNA, aren't they? So if you don't have them, it is going to cause defects in the DNA. UV radiation is a well-known damage to the DNA. Ionizing radiation, likewise. Oncogene signaling, that is important for us. So what I was showing you in the previous session, if you have any oncogene signaling, P53 will become active in cancer cells. Hypoxia is also known to activate. So less concentration of oxygen can also activate P53, and the block of transcription. All of these factors are some of these examples which can allow P53 to become stabilized make a homotetramer, so four copies of the protein come together. Now, P53 is very smart. It does not lead to apoptosis straight away. It does not kill the cells straight away, as you saw in this example, that there was an apoptosis taking place here. In fact, P53 is one of the good genes because it tries to manage the damage. How? First of all, it tries and blocks the cell cycle progression of a cell. So that allows cells, what P53 does by doing so is allow cells to repair the DNA damage. So I was showing you some examples yesterday of base excision repair, mismatch excision repair, nucleotide excision repair, and I showed you there were lots of different proteins, genome maintenance protein, we call them. Many of them, their expression is under control of P53. And you can see here why this is important because by blocking cell cycle, the P53 wants to give time for a cell to repair any damaged DNA. So P53 is, of course, trying to mitigate this damage to the DNA. Not having it can cause cancer, but if a patient is being treated through cancer therapy, for example, radiation therapy, do you want P53 to become reactive? No. So in cases where someone has full-blown cancer, they're being treated by therapy which will intended to damage the DNA, chemotherapy, platinum drugs, radiotherapy. In those circumstances, it is noted that tumor suppressors can become reactivated, causing therapy to fail. It doesn't happen in all patients, but there are different reasons because of which therapies can fail. One of the examples will be this one. There is something else that P53 does is that it blocks the progression through the angiogenesis, formation of new blood vessels, because really what cancer is trying to do is by the pathway I was showing you yesterday through which the cancer migrates from the primary site to other sites is make new blood vessels that allows cancer cells to traverse far away into the body through the blood. So one of the things P53 also does is to block the angiogenesis. And if nothing works, it does not really help the DNA. The damage to the DNA is beyond repair, which is what the radiotherapists intend to do in the hospitals, is to damage the DNA of cancer cells beyond repair. Then the P53 will cause those cells to die. So all these signals activate P53, and P53, you can see this is very important, so important that this is called guardian of the genome. So what happens as a result of when cell cycle is arrested? Some cells might become senescent. They just don't divide. Is it good? Is it bad? Sometimes it may be bad because some cells, they just stop dividing. They think we are being killed because we are dividing. Let's not divide. You know, the therapies, many of them work because cancer cells are dividing very, very fast. They just stop dividing. And once the defect is corrected, they will return to proliferation. And as I said, if the damage is beyond repair, the cells will undergo apoptosis. So when I said that ionizing radiation, anything, any threat to DNA can activate P53, I'll give you a classical example. What happens here? Have a look here in this diagram. P53 becomes phosphorylated and stabilized. In its unphosphorylated form, P53 is not stabilized. It is degraded by MDM2 pathway. You all know this. This is classical textbook molecular biology. What activates P53 is DNA damage, one of the things. And this is DNA damage which is sensed by this protein called ATM. So ATM is a kinase. One of the things ATM does, it senses any DNA double strand breaks and sends a signal to P53 in the form of phosphorylating it and stabilizing it to allow it to carry out the role I showed here, one of these rows here. Now, you might want to ask how many substrates are there for ATM? Is it P53 the only substrate? Well, we know about 800 proteins are phosphorylated by ATM once it becomes active. So you can see how enormous the role of ATM is. I'm only touching on one very brief pathway here, which is ATM mediated activation of P53. And once activated, you can see here what P53 is trying to do. It is blocking the cell cycle by making proteins like P21. If this is good enough, by blocking cell cycles, it allows cells to buy that precious time that will allow the DNA to become repaired. Well and good. But if that doesn't happen, the last resort P53 has is for the greater good of the whole organism, and the cancer does not manifest. They will allow those cells to actually die. Now, we call P53 as being the guardian of the genome. Have a look at this slide. You can see as many as 50% of the women who have this ovarian cancer, but also our population with esophageal cancer, colorectal cancer that Dr. Aziz studies, have a look, 45% of the patients will have TP53 mutations. This is enormous, and that tells you something very important. As long as P53 and other tumor suppressors in other cancers, it is all context dependent. I'm giving you one simple, simplified, should I say, example. As long as they are intact, as long as they are not mutated, it's very hard to get cancer, despite having oncogenes. Because even if there are oncogenes, cells can sense those presence of oncogenes and will allow these tumor suppressors to become active, and in doing so it can actually kill a cell in which there is any which is going to cause any future threat in the form of becoming cancer. Now compare this with an oncogene. For example, lung cancer. Lung cancers is driven by different mutations in different oncogenic drivers. So these are some of the oncogenes. Many of them are kinases. You can see here 25% are driven by KRAS, 17% EGFR, ALK, 5% MET, 3% HER2, 2%, and so on. The numbers are too little. So if you start to stratify, to divide patients based on the expression of these oncogenes, it's going to be very difficult to treat a patient actually, because many of them might have KRAS, but in addition they may have EGFR as well, they might have HER2. It's very difficult. So the cell is using different oncogenes, in this case you can see, to drive cancer growth, a cancer cell. But you can see here as many as 50% women who have ovarian cancer would rely on TP53 to not develop cancer. So if you can see, there is a big proportion of women which are kept safe by from developing ovarian cancer because they have good TP53, and that's why the mutations which cause TP53 do not function are so common. So it does not really matter what sort of mutations you have in oncogenes or proto-oncogenes causing them to become oncogenes. As long as these tumor suppressors are intact, normally a cell is very good at taking care of these threats which come in the form of mutations in the DNA. So I hope that is clear. We still have some time. I will start taking questions after this session, so please bear with me. Going to the next slide, I'm going to tell you what sort of mutations are found in TP53 in bladder cancer. Have a look here. There are all sorts of these mutations. These are mutations in exon. So this is the P53 gene. You can see here, these are the mutations in exons. These are point mutations shown here. These are silent mutations, and these are splice site mutations. But you can see here there are three parts of this protein as a transcription factor. I was giving you example of androgen receptor. Remember, androgen receptor is a transcription factor. So is P53. Why? Because it binds to DNA to drive mRNA expression of specific genes. It drives transcription. What should they have? all transcription factor a domain which is very common is the DNA binding. So they need to bind to DNA in order to drive transcription. So you can see all these mutations which are found in cancer, basically majority of them affect the ability of p53 to bind to DNA. If it cannot bind to DNA, it cannot make those proteins which are needed in order to block cell cycle progression, which are needed in order for the DNA to be repaired, which are needed for a cell to carry out apoptosis. So you can see here how important the p53 is. There are many other oncogenes, but the p53 stands out because of its extensive role in different tumor types. So there are groups like this one who tried to restore the function of p53. Function is lost, p53 function. So they tried to restore to reactivate p53 by using this compound CP53 in skin cancers, and to some extent they were successful, at least in experimental conditions, showing that the treatment administration of this compound to mice actually can reactivate p53 dependent function and saves those mice from getting a worse disease, skin cancer type. So in fact you can do that, but you can only do that if you can ask yourself, if you Google this, how successful has this been? You can only do that if there are mutations which you can mitigate by this drug. Sometimes you might find there is a loss of expression of p53 due to the genomic loss. So the gene altogether is mutated, is gone. Basically, what you can restore them? Nothing. But most of the time, you have these mutations, and if you have compounds like this, which can actually restore the function of protein, which is good news, in order to make treatment work better. Which kind of treatment? Not radiotherapy, because radiotherapists, they're trying to overburden cells with an enormous amount of DNA damage beyond repair. And if you activate p53 in them, it might allow cells to actually get DNA repair pathway activated. You don't want that. So this is certainly very important. There is a question I can see, and Sandeepan, I would have a slide. Sandeepan is asking, it was seen elephants have more copies of T53, making them immune to cancer. If by any means we try to mimic the same in cancer by birth, it will be viable in the first place. Sandeepan, I'm coming to your question. Have a look at this slide, Sandeepan and everyone. It looks like P53 keeps animals safe, such as elephants, these giants, from developing cancer. Um, why? And do they live any shorter than humans? Of course, we get cancer because we live long enough. We have lots of cell division. We saw that yesterday. The cells are dividing much more faster. In some tumors, they have more propensity of developing cancer. Some cells don't divide at such a fast rate. The cancers are rare, rather. In this case, animals actually live as long as average human. Despite that, there are no cases reported in humans. You can see here 25% of humans will develop cancer, while elephants, they don't develop any cancer. It turns out they have many copies, as many as 19 to 20 more copies of p53. And as a result of this, they are kept safe from developing the cancers. They just don't develop it. You might ask, okay, how do you put an elephant under a scanner, an MRI and a scan to see whether they're developing cancer? Well, not every human being would go to MRI scan to prevent them from cancer. You go to a doctor when you have any problems. Elephants don't get those sort of problems that will be obvious indications they are actually developing cancer. Some of them can be detected by blood-based assays as well. So in fact, they don't develop cancer. And what happens here is, Sandeepan was asking this question that can we try this approach in human? Well, it might be that if there are some humans who, as an anomaly, defects in the genome, may have more copies, may actually survive longer may be immune to cancer, but that's usually not the case. You don't have very many copies of p53 in humans. So why elephant? Nature gave elephant. I thought about this question a lot. Why nature gave elephants so many copies of p53, not to us? And I think, and this is my personal view, I think the answer is that elephants are really, really huge organisms. They can afford to have a lot of cells undergoing apoptosis. That is one of the primary functions of p53 in embryonal stages in normal non-cancer cells. They can carry out apoptosis. If you have 20 times more apoptosis in an elephant, what is happening? They may still withstand this extensive loss of cells. Humans, as opposed to elephants, are not so big. They may not be able to be saved from this so much loss of cells. They might actually become um, uh, different type of illnesses, have developmental disorders if nature gave us so many copies of p53. So I would say that one of the arguments is for our own good, so that we don't have atrophy of different organs. The nature did not give us so many copies as it did to elephants. And of course, there are other differences like diet and so on. They don't eat junk food, do they? It's like us. So, and many other things, of course. So I hope that will answer the question.
Can I add here something,
Yes, please. Yes,
Rasan?
please. Yes,
Uh,
please.
this is a very nice observation, and there's another one that happens in Wales. The
Uh-huh.
Wales, they have got only one copy of p53, and they live for hundreds of years, probably more than 100 years, and still they don't
Uh-huh.
develop cancer. There are no, known cases of cancer in Wales. So it was found that p53, very good. It's a guardian of the cells, and it's holding back the tumor suppressive gene. But whales have got a very good and rapid mechanism of DNA repair, because there the cells are constantly dividing, and they have to keep up with their energy as well. So they have got a lot of faster processes that are going on. Probability of having cancer with the way in the whales was much higher than in elephants, and they have got only one copy of p53. Still they don't develop cancer. So
Okay.
there are alternate mechanisms that are keeping them safe. That's what I would
That's an
like to add
excellent
here.
that's a very interesting and excellent point you've added. I think if they don't have very much p53 and they have very robust, much robust system for repair of DNA, I would think, am I correct that they don't develop as much variations as humans do? Because part of the DNA that we develop mutation is to make us more adaptable and evolutionary successful. Uh, is it possible, is it the case with whales that they have a lot of adaptation abilities, or they have a very small population which is very genomically synchronized, that
Yeah.
they don't adapt, they don't evolve perhaps? So there is a trade-off, I think. If you have a system which allows you to repair DNA very, very quickly, you may not actually evolve at all. Like some fishes I know, they remain alive for 3, 4, 500 years, and there are no changes. They don't evolve at all as a result. So perhaps they have a very excellent, I would say, DNA repair system, but then it comes at a cost of them not being evolved as humans have. Who knows?
Yeah, this again reinforces the concept that every species is unique, every species has got their own
That's right.
mechanism. Let's deal it that way. So, interesting discussion. I think anybody else would like to jump in and add to it. Thank you.
Okay, so I will, if there aren't any, then I can carry on with more questions. We will start taking some questions. Padmavati, I see there is a question. Let me quickly finish this if I can. Have a look at this paper. I was showing you this paper yesterday and it came back again. Antioxidants accelerating, they're promoting antioxidants which we buy in the supermarkets, especially in these countries in the West. Antioxidants, they're good for you. That's what we have been made to believe and deceive. Basically, this is one of the excellent examples, this publication showing that antioxidants used in this paper actually can degrade p53. So instead of stopping cancer, they can actually accelerate cancer. So uh, my personal view is I don't take any antioxidant supplements. I get it from my food, my fruits, vegetables, um, and so on. So that would be one example of it. So the other disease, somebody was asking yesterday, Dr. Aziz actually himself was asking about retinoblastoma. You can see here this disease. And I should tell you something. There are cancers. Sometimes I think there are lots of cancers which we can treat with where the treatment is available. We know there is a standard treatment, but because of the healthcare system, the infrastructure, those treatments are not available to human beings at par with every other nation. So have a look here. This individual, this child, which was identified to have this disease, retinoblastoma, at much earlier in Canada, and was successfully treated before, sadly, a child in Africa where the tumor actually became full bone, the retinoblastoma invaded into the brain. Which is a bad news, of course. So you can see here from the initial diagnosis, very early stages to very late stages, the cancer can actually progress. But the treatment is very successful here because it's a childhood cancer, which is mostly germline related, meaning they inherited bad genes from their parents. They were not as a result of some mutations in the children. They will not have the possibility of those sort of mutations happening in such a short lifespan that they have lived, but they actually inherit, sadly, germline mutations, bad copies of the um, proteins, genes from the parents. So basically, this is how it is formed in the back of tumor here, back of the retina. I'm sorry, and you can see here this can actually manifest itself to such horrendous conditions when it is fully grown. And you can see here there's another transcription factor that regulates this, and there is another tumor suppressor that regulates this formation of this tumor. We call it retinoblastoma. This is mostly in, you can see in childhood cancer, this is very common. And you can see here what is happening. P53, we saw was becoming active as a result of phosphorylation by ATM in the charts I showed you previously. So phosphorylation was making p53 active. It was able to form tetramer, bind to the DNA. In this case, what is happening is this tumor suppressor is being regulated differently by cyclin CDK. Now, these complexes are very important. Cyclins are proteins which regulate the entire cell cycle of cells, as you know. So what happens is the cells decide to divide. They have transcription factors that allow them to carry out this. And one of the most important one is called E2F. So E2F is a transcription factor. If I ask you what is the most common domain you will find in a transcription factor, is a DNA binding domain, right? So it cannot bind to DNA every time. Why? Because it is being trapped, it is being stopped, it is being inhibited by this protein called retinoblastoma. It binds to it tightly, and by doing so, it stops the E2F from becoming active. Dr. Aziz, shall we take a break and then I can conclude this session, or do you want me to carry on? I have a couple of more slides and that's it.
Well, yes.
And then I can take questions. I just don't want the students to become overburdened. Um, done 45 minutes. So let me finish this anyway, if there are no questions. Is that okay?
Because this will be
Okay.
followed by a tutorial, right? So
That will be then the tutorial. I have one more slide and then we can
I
start
think
the
let me
tutorial.
give a break, and then
Okay, we can finish this
the
session and then
tutorial
we'll
is
move
going to
back
be
on.
for 2 hours. So you can take your
Yeah,
coffee.
I understand. Yeah.
Uh, so to
So
all the
shall we meet at,
participants, yeah,
so let me finish this after this
okay,
session, and then in the meantime, I can answer the questions which
okay.
were being asked. Perfect. Okay, so Let me have a look.
So shall I send you some coffee?
No, thank you very much. I don't drink coffee. Uh,
Okay. So you have Cafe Nero and Starbucks both, right?
I,
Yeah.
I I don't think when was the last time in the last decade I had to go to. If I'm on an airport, I might have some tea, but that's not really the tree. You need to boil the tea in the hot water to make the real chai. Uh,
Yeah.
yes, so there are questions I can now attend to. Padmavati is asking, we observed such increase in cell proliferation at a lower concentration due to phytochemical screening. And okay, that's true. If you use a suboptimal doses of uh, some drugs, actually you can see an increase. That's true. So that's why it is very important to titrate and establish a maximum tolerated dose that you can give to a patient without causing much severe side effects. So certainly that is a good observation, Padmavati. Khalida is asking, cancer caused by, okay, and this has been answered already. Uh, Sandeepan is asking, I believe Sandeepan, I've answered your question, and Dr. Aziz as well. Saida is asking, okay, let's see, admin. Padmavati is asking, while it's naturally present in elephants, why is it mentioned as a transgene, sir? What does it signify? Very good question. On top of the head, I don't remember. I think this because this is not really a CP53, meaning a cellular, although it is present in the cell. It is in terms of its regulation. There may be that it is constitutively active. I know that this P53, which we call transgene, actually does carry out DNA repair. So the function is very analogous. Why the different name? I'll have to go and check. But from what it looks like, from what I know, is that it is able to carry out analogous functions as does the cellular P53 by safeguarding the genome. It's just a different name, perhaps because it is a product of transposons, making more copies, amplifying the genomic locus where this P53 is present. Perhaps that is what is signified by the name transgene. So I think that's pretty much it. Shall we join in 10 minutes?
Yeah, sure.
Okay. I'll see you soon then, everyone. Please feel free to keep on posting your questions so that we can discuss them before we jump onto the omics.
Would there be any need for tutorials? Oh, sorry.
Sorry, go on.
Do they need a different
Sure.
system or any other apparatus for the tutorial?
I
So that they can
think
get more?
so. I have some slides
Okay.
that I can first go to, and then I can show them some platforms because it is being recorded in their own time. They can go through some of these platforms and gain the experience that they need. Somebody was asking, I think Khalida was asking about computational biology.
Mhm.
Of course, I think it is an excellent career. What I do suggest people who want to do computational biology as a career, they should be really really smart in mathematics. Without it, they will not be able to efficiently code, and some people are really super smart in mathematics, and actually do develop to become a good computational biologist. We in fact need much more computational biologists now that we have a lot of data, astronomic data coming from different labs. We can sequence, yes, but how do you make sense of this large genome sequence data from 3,000 patients? You need good computational biologists to analyze, make sense of that data, uh, and those are the individual scientists which will be in much higher demand than anyone else. So one needs to think about that. But yes, please do see where your passion lies, because what we do, Dr. Aziz, myself, you can still, as you can see, become an academic if you have a passion for wet lab science, because there are lots of wet lab scientists these days. But computational biology is a very fast developing area. You need to have the right skill sets, and inherent interest in mathematics, coding, in order to become one. So Arham is asking a question. A new drug is made which takes
I can take that if
approximately.
you allow me, and
Yes,
you can
please
take
do.
a break. Yeah.
Yeah. Okay. Thank you.
So, Arham, it's not about inventing or discovering a new drug for every person. It's about matchmaking. The drugs and molecules and biologics, they are already available, and there are millions of them. What we have to do is to find the right drug for the right person at the right time. That's what we mean by personalized medicine. Don't get confused. This is a very common confusion that we will be knowing the target, and then we'll be developing 10 to 15 years, or even if it is accelerated for a couple of years to develop a new medicine. No, the molecules are already there. We need to get give it to the right person. Okay. If that answers your question, otherwise you can further ask, and Dr. Asim can enrich that as well.
Any other questions?
No, I don't see that. It's still time, 5 minutes. I would encourage the participants who are at that stage, they can also ask Dr. Asim about their career-related questions, like somebody did about the career in computational biology. But if you have other questions regarding the internships or PhD positions or postdocs or scientist positions, I think Dr. Asim would be able to give you an overview or some some guidelines, some pointers that will be helpful
Yes,
for your
please
career.
do. Yeah,
Yeah.
I'll be
So,
delighted
from
to.
yeah, apart from the science, utilize this question answer session for other things as well. Okay, thank you.
Okay, that's great. So, shall we resume and finish this before we switch to the omics tutorial for today? And as Dr. Z said, please feel free to ask me. Open your mic if you want to show yourself. Go ahead and discuss if you have any career-related queries. Um, Okay, so what you're seeing here, what is happening in this disease, the cancer of retina, is basically one single gene mutated sometimes, and what it is the loss of function mutations in retinoblastoma, which I said is a tumor suppressor. So normally this pathway is regulated highly by and coordinated by the cyclins. So cyclins needs to become activated by binding of the cyclin CDK complexes. And one of the activation signal is the loss of P50, P16 protein. And when this stops binding to this trimer, the dimer becomes active. So you can see how much variety is there. Sometimes proteins might become active as a monomer. Sometimes they need to be tetramer. Sometimes they need to be dimer. Sometimes they need to be phosphorylated, sometimes they need to be dephosphorylated in order to become active. And all it lies is in the changes in the conformation of protein that results from such complex processes. And see what is happening here. In the when the cells decides to divide, and before that, of course, the DNA needs to be replicated. So what happens in a G1 phase of cell cycle is that complex becomes active, and the activation simply means that now this kinase can phosphorylate a retinoblastoma, but it is only happening in G1 phase of cell cycle. And this phosphorylation actually makes a retinoblastoma inactive, this phosphorylated retinoblastoma. And that allows E2F to become active. When E2F as a transcription factor becomes active, uh, what happens is there is expression of genes. Which genes? Genes that are important for DNA replication. In fact, DNA polymerase, we discussed this previously, the proofreading function, if you remember. DNA polymerase, the expression is under control of E2F. So for DNA, for a cell to prepare for a division, it needs to have the entire DNA replicated. And thus, this replication requires these proteins, cyclins. And these proteins, how do they do that? Is by allowing E2F to become active. How does it become active? When a retinoblastoma is phosphorylated. So remember, it's the same name of the protein or the gene and same name of the disease, retinoblastoma, as I showed you on the previous slide. So what is happening here is retinoblastoma needs to be phosphorylated by this complex. And when this complex becomes active, retinoblastoma is phosphorylated. E2F can now drive the transcription of genes, which we need for DNA synthesis. And it has been found that there are germline mutations, meaning mutations that passed on from parents into the little poor child to have mutations in retinoblastoma. What sort of mutations? The loss of function mutations. And as a result of these loss of function mutations, retinoblastoma cannot efficiently capture E2F when it should not be active. And as a result, E2F is always on in both conditions. And remember the switch I was showing you. And once this is on, you can imagine this can lead to diseases like we discuss here, diseases like retinoblastoma. In Western countries, hardly anyone dies of retinoblastoma. Do you know one of the reasons? It is early detection. You can detect the cancer much earlier than it reaches to this stage. But also, retinoblastoma does not have a lot of mutations. Usually, the mutations which you find in a retinoblastoma are restricted to the RB gene itself. And these mutations are only handful. They're not very many. And there aren't other oncogenes, there aren't other genes which are mutated, only this one. And this is why the mutational noise of this cancer, meaning how many background mutations are there in a 3 billion genome, they're very little. They're normally restricted to one gene. So the mutational overall noise is much more restricted. You will not send a patient with retinoblastoma for whole genome sequencing because you know that most of these mutations are restricted to just one single gene. And therefore, because their mutational profile is super restricted, the mutational noise is very, very little, these tumors tend not to metastasize because metastasis, as we discussed yesterday, requires a whole new set of different mutation happening at different times of a life cycle of a cell and its products that will take a very long time, and that's why cancer diagnosis is usually very common as we grow older compared to children. So you can see there is an element of mutational burden. If there is a high mutational burden, like in many diseases, prostate cancer, breast cancer, compared to this one where mutational burden is super, super low, you can see that the treatment of such cancers will be much more straightforward. The prognosis, the outcome of the therapy looks much more positive if the mutational burden is very, very low. Compared to those cancers where there is an extensive changes to the genome, how do we identify those changes? One of the things is to do RNA sequencing that Dr. Aziz discussed earlier in the day today. So that is one aspect I wanted to touch. So to summarize this, you can see here there is a cooperation going on in a normal cells. There is a regulation where proto-oncogenes are promoting growth. They can reverse injuries. In terms of having injuries, of course, proto-oncogene functions become heightened compared to the normal levels. And tumor suppressor genes are blocking any unregulated growth. But what happens in a cancer cell as a result of mutations, which mutations, gain of function mutations in proto-oncogenes, which transform them into oncogenes, and loss of function mutations into tumor suppressor genes, which allow them to lose their function. If you combine the effect of this, the cell will have an overall abnormal growth. And that is the first step on a cancer pathway. So they will have a lot of barriers to cross. We discussed a lot of these barriers yesterday in the sessions we had, and that's why those tumors which actually metastasize to distant organs, they will need to have a lot of mutation burden. They will have a lot of mutations in very different pathways to carry out this journey from the primary tumor, dissolving the membrane where the epithelial cells are sitting, moving into the connective tissue, gaining access to the bloodstream, surviving in the bloodstream, surviving the immune system, then going and getting out of the blood vessels, trying to find the right organs, traversing through those organs and making their colonies. So they need to be super fit to carry out these journeys. But that comes at a goes to the host organisms, and that comes as a result of a number of mutations which make them fit. And that's why you will have a much heightened mutation load in tumors which are metastatic compared to the tumors which are still at primary site. So in a disease, it is possible a liver cancer or a colorectal cancer, if you open up a patient and have a look at the primary tumor in large intestine colon, you might find the mutation profile is rather restricted compared to the same colon cancer which has gone into liver. They may have, in addition to the primary mutations in the primary cancer, they may have a new signature of mutations that allowed them to invade and metastasize into the new organ and sustain their colonies there. So mutation overload